And from this competency is very important. You can expect a long type of question on describe the renal regulation of acid-base balance, direct question, or the mechanisms involved in pH regulation by the kidneys. Okay. Beside this, there may be some short questions, explanatory type of questions, and these are, the first two is almost same. And there are other questions like acidosis. Two types of acidosis can happen. One is respiratory acidosis, another is metabolic acidosis. Similarly, because of imbalance in pH regulation, there can develop alkalosis. And again, the alkalosis are of two types. One is metabolic and another is respiratory. It means by respiration, by suppressing breathing, respiration, or by increasing breathing, we can imbalance the pH regulation. We'll describe how. Also, during metabolism, we produce various acids through metabolism. And these acids are volatile acids and non-volatile acids. So before coming into volatile and non-volatile acids, let us see what you mean by acids. So what are acids? Acids are what? Compound substances that can donate hydrogen proton. That can donate proton means hydrogen ion. Hydrogen ion. So they are the substances that can donate hydrogen ion or proton is called acid. And the opposite is who can accept this proton, accept these hydrogen ions, they are called base. So as I said, during our metabolism, we are producing lot of hydrogen ions, lot of acids. These are, as I said, volatile acids and non-volatile acids. What are non-volatile acids? Volatile acids are formed by carbon dioxide. Okay? When it joins with water, it forms an acid, H2CO3. And when it dissociates, it produces hydrogen ion. Okay? So by this mechanism, volatile, carbon dioxide is a volatile gas. So by this mechanism, either by increasing the production of carbon dioxide, we can increase the hydrogen atom. By decreasing the carbon dioxide level, we can decrease the hydrogen atom. Okay. So in this way, by modifying the volatile acids, we can balance acid-base balance. So this is one point. And volatile acids, or non-volatile acids, see, in our body continuously, carbohydrate is breaking down to provide us energy. Okay? So during these processes, particularly in anaerobic condition, we produce some acids. What are these acids? Lactic acid. So this is an acid, non-volatile acid. Similarly, during metabolism, fatty acid metabolism, we produce lot of keto acids. After beta oxidation, lot of acetyl CoA will be formed, and some will be oxidized in the TCA cycle to give us energy, and some will be converted into keto acids. Can you name such keto acids? Acetoacetic acid, alpha ketobutyric acid. So these are keto acids. This is observed in diabetic mellitus patients. Why? Because these patients, because of absence of insulin, the body relies on, it cannot depend on carbohydrate metabolism, so it has to depend on fat metabolism. As a result, there will be lot of production of ketone bodies, acetone. If the urine of the diabetic patient is tested or smelled, you would find a sweet smell. And this is because of acetone, one kind of ketone bodies. Okay, so it is frequently seen in this condition. Or when you are exercising, vigorous exercise, what will happen? You will first utilize your stored carbohydrates, then after some time it will be exhausted. Then which nutrient you will be depending on for deriving your energy? Fat. Okay, so fatty acid will be burnt. As a result, lot of these keto acids may be formed. Okay, so these are another source from carbohydrate metabolism and from fat metabolism. We can get some from some base acids. Similarly, from protein metabolism, we can get some acids. Okay, what are these acids? What are these acids? Amino acids, ultimately, lot of say when I am doing exercise, what will be there? Sources of, or I am fasting for a prolonged period. So what will happen? You try to derive your energy from carbohydrates, but storage is exhausted. Then you will rely on fat. Since it will produce lot of ketone bodies, so body will try to derive its energy from the protein, body protein. Okay, now this body protein will be breaking down into amino acids. Now these amino acids will undergo, first it will undergo deamination, transamination. So it will form some keto acid, pyruvic acid. From that pyruvic acid, lactic acid may be formed, oxaloacetic acid. So this way different acids can be formed. When these are excess in blood, this can be formed. Okay, so this way body is generating a lot of hydrogen ions by different metabolic processes, oxidation of different metabolic processes. Not only this, nucleic acid. When a cell is become dead, then what will happen? The cell is having DNA, RNA that are containing nucleic acids. So this nucleic acid will come out. And this may also can increase the acidity. Okay. And why this can happen? It can happen through this, this nucleic acid, ultimately it will produce purine, and from purine, metabolism will get uric acid, another acid. So this is another source of acids in our body. Since lot of acids are produced, our body will become acidic. Means our body pH will not be alkaline in that condition. It will be acidic. Less than 7, it will be coming down. Serious condition. So it has to be prevented. It has to be, this accumulation of acid has to be prevented. Who will be preventing? There are some mechanisms. Similarly, by suppressing your breathing or increasing your breathing, what can happen? How you can change the hydrogen ion level in your blood? Madhurima, I am suppressing my breath for some time. What will happen to the blood? Whether acidity, the blood will become acidic or blood will become alkaline. You are not breathing. That means carbon dioxide is accumulating in your cell. What will happen to this carbon dioxide? You are not breathing. Breathing is suppressed. You are unable to breathe out the carbon dioxide into the air. So there will be accumulation of carbon dioxide. Now, what will happen to this carbon dioxide? It will join with water and will form H2CO3, carbonic acid, okay, with the help of the carbonic anhydrase. Now, this will be dissociated into hydrogen ion and bicarbonate. So, when carbon dioxide level is increased, pH will be decreased, because pH decrease means more concentration of hydrogen ion. Okay, more concentration of hydrogen. When there is less alkaline, less concentration of hydrogen, and that means the condition is becoming too alkaline. Blood is becoming alkaline. So, by suppressing breathing, there are several conditions in which breathing can be suppressed. Okay? Voluntarily you can suppress your breathing. Isn't it? There are some conditions. There is some pathological condition. Obstructive pulmonary disease. Breathing is suppressed. So there you can develop acidosis. Increased production of hydrogen ion. Similarly, now I am doing this thing. Hyperventilating. Or when you became very angry, emotionally excited, what happens? Our breathing rate is increased. What can happen? What can happen? More carbon dioxide is washing out. Less production of hydrogen ion. Means blood is becoming basic, alkaline. So by altering our breathing rate or suppressing the breathing, we can change the hydrogen ion concentration of blood. If it is changed, it can happen in the cell. So these changes, hydrogen ion changes, can increase, decrease also within the cell, means in the body fluids. So this is one mechanism. Okay. So, what to do? We must control it. We can see we are having our pH very well regulated. This is the homeostasis. Homeostasis, one criteria of homeostasis. It has to be maintained, pH has to be maintained at 7.4. It will vary. There is a normal range. What is the normal range? 7.35 to 7.45. If the pH goes beyond 7.45, what will be? It is alkalosis or alkalosis. If the pH comes down to down below 7.35, the condition will be acidosis. So to prevent alkalosis and acidosis, what has to be done? It has to be controlled. So we have some controlling mechanisms. Okay, what are these controlling mechanisms? Okay, these are the controlling mechanisms. One is blood is having some buffer system. Also, the cell is having some buffer system to maintain the pH within the cell. Blood is having some buffer system to maintain, to control the ups and downs of pH within the normal range. And our respiratory system is there to maintain the blood pH. If it is maintained, it can alter it, it can maintain it. Okay, if it is maintained under altered condition, if it is maintained by the respiratory system, then the blood will also try to maintain the pH of the extracellular fluid, interstitial fluid, or intracellular fluid. Okay, so it has got, and this process, it takes only seconds to buffer. When it is altered within second, it can change almost overtime. But their capacity is less. Their capacity, how much it can change? Their capacity is less, but they are very fast. It is performed in seconds. And the second one, it takes minutes. It takes minutes to control the pH. Okay? And the renal system, how much time it will take? It is the most efficient, vital part among all these. It is the vital organ. Kidney is the vital organ in controlling this. How much time it can take? Days, months. Very slow process, but very efficient. Okay, its capacity is much high. Okay. Now, let us discuss this. Blood buffer system. Okay, what are they? Bicarbonate buffers. Okay, let's see. How bicarbonate buffer? Bicarbonate buffer means NaHCO3 NaH2CO3 or HCO3 H2CO3. This is the buffer. Are they okay? Okay. Now what happens? There is excess hydrogen ion came from various sources. Now how it will buffer it? One of the component of this buffer, they will come into action to neutralize those hydrogen ion. Very simple. This will combine with that hydrogen ion. So what will be formed? H2CO3. Oh, you can't see. H2CO3 will be formed. Now this will be broken down into carbon dioxide and water. Gone. Problem solved. Okay, this way we can neutralize the hydrogen ion, the acid. When blood became what? Alkaline. Means there is less hydrogen ion. I need more hydrogen ion. How can? This H2CO3, this component H2CO3, it will be dissociated into plus H plus. So this hydrogen came into solution, it will decrease the pH, increase the hydrogen ion. Problem solved. So this way, the buffers present in the cell, buffer present in the ECF, this buffer present in the blood, they can neutralize the excess acid. Okay? But their capacity is again limited. Okay. Now, the phosphate buffer. Phosphate buffer is present. Now tell me one thing. Phosphate concentration, whether it is high within the cell or outside the cell, means outside the cell means extracellular fluid, where it is more. When we had been discussing about the development of resting membrane potential, inside is negatively charged, more negatively charged. Why? Phosphate, they are easily not permeable, so they remain within, and proteins negatively charged. Okay, chloride is outside more, inside is less, but here these ions are more, relatively more. Though potassium ion concentration is more inside, but ultimately in relation to exterior, inside is negatively charged, particularly along the membrane, inner membrane, inside of the membrane, that is negatively charged. So phosphate ion is predominantly located within the cell. So it is a good buffer for maintaining the pH within the cell. Also, they are present in glomerular filtrate. These phosphates are also present in glomerular filtrate. We'll see they also take part in acid-base regulations. Another process is there. Another buffer is there. Okay, let's see how phosphate buffer acts. One component. Another component is NaH2PO4. Now this is the phosphate buffer. Now if I add hydrogen ion to make the fluid acidic, now how they will neutralize it? Hello? First, second, third, fourth brains. Either you or you. Can you tell me how the phosphate buffer will neutralize the hydrogen ion? You are intelligent, that's why I am asking you. Intrusions. I am not joking. Okay. So, about your performance, when you asked me some questions, you inquired about your results, why it came so, then I understood, yes, you have some material. Okay, so that's why I'm asking you how you can neutralize this acid. Anybody? Anybody? How this can be? Which one is acidic? This one, which one is here? There is one weak acid. And a salt of weak strong base. So here, which is acidic? Whether this one or this one. This can donate one of the hydrogen ion. So definitely, this is acid, this is already it is having two hydrogen atoms. So it will not take part. Who will take part then? This. So this will become H2PO4 and this hydrogen, this will be released. Okay? So this way it can buffer, the phosphate can buffer the hydrogen ions. Okay? Then another buffer is there, protein buffer. Can anybody tell me one of the function of plasma proteins? It among various transportation and all of so many functions plasma proteins are having. One of the function is its access buffer. Isn't it? Can you tell me how plasma protein can help in buffer? Interesting. Can anybody tell? Proteins are made up of amino acids. Okay? Amino acids. So, R1, R2, these are amino acids, no? R3, in a peptide chain, R4, and at the terminal, suppose you are the chain, and you are interlinked by peptide bonds. And every amino acid is having one amino group and one carboxylic group at least. So one carboxylic group you have used with his amino group to form the peptide linkage. What happened to your left hand, right hand? It is what? Either it is carboxylic group or amino group. It is amino group. Similarly, that girl, they are in a peptide chain. Her left hand will be opposite, that will be free. If it is a free amino carboxylic group, that will be free amino group. Now this will participate in acid-base rise, the terminal amino acids. How? So this is one amino acid, say terminal 1. Okay, it is having free carboxylic group, and another terminal is having NH2 group. Now I have added hydrogen ion. What will happen? This cannot be accepted by this. It can accept and can become NH3 plus. Isn't it? It is buffered. Now the solution is alkaline, basic. Need more hydrogen atom, hydrogen ion. So what to do? This can donate one hydrogen atom, hydrogen ion, and this will become COA negative. In this way, protein can, plasma proteins, they remain in plasma, so they can maintain the pH buffer, the excess hydrogen atom, and when there is need, it can also release hydrogen atom. But so that way it can maintain pH. Right? Okay. Now, what happened? Can RBC can do it? Can RBC can do it? It can be done by the RBCs? How? It is also having a protein. What is this protein? Hemoglobin. Globin is again a protein. So there will also be in the programming chain, 4 chains are there. So every chain is having a terminal amino group, terminal carboxylic group. And some amino acids are having not only terminal, within the amino acids, some are having more than one amino group, more than carboxylic, they can also participate. Okay, so in the same manner, so within the RBC, the hemoglobin, we will find, we have asked what are the functions of RBC. So that you can tell, yes, RBC can, besides transporting gases, carbon dioxide and oxygen, it can also act as buffer. I think the first point is explained, you understood it. Correct? Okay. Let us, respiratory system I already told. How it can increase the hydrogen ion, how it can decrease the hydrogen ion? By increasing or suppressing the breathing. Very simple. Okay. And the last renal system, okay, and that is our today's topic. I already spent 10 minutes. I started late by 10 minutes. So we are already half an hour gone. Another 20-25 minutes I am having. So I'll try to explain that part. Our kidney has three mechanisms. How kidney balances the pH. How kidney controls the pH. It has three mechanisms. All these three mechanisms, purpose is what? All this mechanism, their purpose is to retain bicarbonate ion so that it is not lost through urine. All the bicarbonate ions has to be retained. Means they are to be reabsorbed from filtrate because lot of bicarbonate ion is filtered. If all these are filtered, go through the urine, our pH, body pH will be jeopardized, will be disturbed. It will be very difficult to maintain the pH. So I need all the hydrogen bicarbonate ions to be reabsorbed. So all this system will be involved in this reabsorption of bicarbonate ion. And another purpose is to secrete hydrogen ions. So two purposes, reabsorption of no tubular cells, only the filtrate will contain bicarbonate ion. No tubular cells will secrete any hydrogen ion, bicarbonate ion. But hydrogen ion, lot of bicarbonate ion is filtered into the lumen, as well as the renal tubular cells also secrete hydrogen ion into this. And my purpose is to absorb all the bicarbonate ions and at the cost of hydrogen ion. And this will be done, we'll see, this will be done in exchange of hydrogen. Hydrogen ion will be secreted into the lumen, or it is already there. Now this hydrogen ion will be utilized for reabsorption of bicarbonate ion. Okay, now we'll see one by one. Okay, how they perform this. Our urine pH, is it acidic or alkaline? It is very acidic. Who made this? Acidic? All these three mechanisms. They involved it. They released, they absorbed all the bicarbonate ion that was filtered. Some hydrogen ion is utilized, some are not. Some part is not reabsorbed, hydrogen atom are not utilized for absorption of or not utilized in the mechanism of absorption of bicarbonate ion. So that part, retaining part, around 4,300 milli equivalent of hydrogen atoms are produced. Are out of this 50 to 100 milli equivalent is excreted and make the urine acidic. So all the out of these 4,328, from this you just subtract this 100 to or 50, then remaining part is utilized for reabsorption of bicarbonate. And this bicarbonate is essential to make a alkali reserve in the body so that it can, within the body, in the body fluids, it can buffer the excess excess hydrogen produced in different ways. See, this mass of bicarbonate ion is filtered throughout the day, and all of these are reabsorbed at the cost of hydrogen ion. Okay, without the help of the hydrogen, this is not possible. And about 4,380, as I said, hydrogen ion is produced. And out of these, this mass is utilized for absorption of bicarbonate ion, and only 50 to 100 milli equivalent is excreted in the urine and make the urine acidic. Let's see this mechanism. There are three mechanisms. One is bicarbonate mechanism. One is phosphate mechanism. Another is ammonia mechanism. So this is the bicarbonate mechanism. What happens? In the tubular lumen, lot of alkali bicarbonate is passing. These are the bicarbonates. They are dissociated into sodium and bicarbonate. Sodium and bicarbonate. And this sodium are what happens within the cell? These cells, whatever it is, the luminal cells, or almost in all cells, they are highly metabolic. They have to run some pumps, isn't it? So lot of carbon dioxide will be produced. Lot of carbon dioxide will be produced. So since energy is produced, carbon dioxide will be produced. Energy is ATP is needed for pumps, various pumps. So as a result, there will be production of carbon dioxide. Beside this carbon dioxide, cellular carbon dioxide, some carbon dioxide also come from tubular lumen. How this will be formed in the tubule? How this is formed? I will come to later. So here within the tubule, these two, water and carbon dioxide, combines together within the cell. This surface, apical surface, is having one enzyme. What is this? Carbonic anhydrase. So under the influence of this carbonic anhydrase, this is formed. Immediately it is dissociated into hydrogen ion and bicarbonate ion. This hydrogen ion will be pumped out. Into the lumen. So this is the pump. This will be utilizing energy. This will be pumped. Now what will happen to this? Some alkali is coming. This will be dissociated into sodium and bicarbonate. Now this sodium, this is what? Sodium, hydrogen, antipore. You, hydrogen, you go, let me come in. So with this antipore, sodium is entering and this hydrogen is going out. Okay? Now this sodium is coming out the interstitial there into blood, and bicarbonate is also coming out, and they again form NaCO2. See what happened? This base, bicarbonate, which was passing through the lumen, it is now through this tubule, it has come into the interstitial fluid, again into the blood. So it is preserved. Okay, so it is preserved. And how it became possible? Because of this hydrogen ion. This hydrogen ion combined with this marker to form H2CO3. And this is dissociated into water, and this carbon dioxide, luminal, this is produced in the lumen. Again by this carbonic anhydrase, it can affect this reaction also, this reaction from both sides. That's why it stays in apical region. Okay, so now what will happen? This hydrogen ion combining with this H2CO2 carbon dioxide is coming in. Again it will form. Now see, this bicarbonate ion is converted into carbon dioxide. This carbon is converted into carbon dioxide. So this carbon dioxide is again forming carbonic acid and bicarbonate ion. This is coming. So ultimately this bicarbonate ion that was present in lumen, it is coming into blood. Reabsorb. At the cost of this hydrogen atom. If it is not helping, then it will not be possible to reabsorb this bicarbonate ion. That's why the tubular lumen should excrete hydrogen atom. Okay? There are two ports, channels that is active during this. One is already discussed, antiport. Another is proton pump. What? Proton pump. It is also there in stomach. During hydrochloric acid synthesis, we have seen this proton pump is there by who are having lot of the acidity problem. So they are given why? To block this, so that hydrogen ion is not released. So here also we'll see another pump is operating. What is this? Proton pump, ATP driven. So both of these require, because again they pumping here, don't, if this will come alone inside, it will come from lumen within the cell without alone through a pump. But in that case, sodium and through the antipode, though it is also a energy dependent. So these are present where? These pumps, these receptors are, these channels, they are present in where? See. Sodium hydrogen pump, they are present in distant columnar tube, and ATP driven proton pump are present in DCT and collecting duct. And these are the three mechanisms of neutralizing or absorbing bicarbonate ion or neutralizing hydrogen ions. Okay, what are these three mechanisms? Bicarbonate mechanism already explained. Now we'll discuss about the phosphate mechanism, then we'll discuss about the ammonia mechanism. So three mechanism, bicarbonate mechanism already told. Now this is the bicarbonate mechanism itself. This is the phosphate mechanism. See, phosphate, our glomerular membrane, they are secreting phosphate and in the form of disodium hydrogen phosphate. So in the lumen, it is dissociated into sodium and sodium hydrogen phosphate. And in the process, in the similar way, what will be from hydrogen ion and bicarbonate will be from this will come out, and this is what? Proton pump. This is what? Proton pump. So with that help of this proton pump, this will come out. Now this sodium hydrogen phosphate will combine with this, and this will be NaH2PO4. So this has become, this was Na2SPO4 less, it is basic thing, but it is the acidic thing. Okay, now it is excreted. So some hydrogen ion is excreted. Okay, so this is the which mechanism? Phosphate mechanism. You can observe it in two places, where within the cell and in the renal tubule. Okay, another mechanism left, that is ammonia mechanism, that is present only in collecting duct. What is this mechanism? Ammonia comes from what? You said from deamination. So within this cell, within this epithelial cell, there is glutamine. Okay, there is also glycine. Some amino acids are when they are deaminated, what they form? Ammonia is formed. Now ammonia can easily enter into. These are water soluble. They can easily penetrate into the luminal fluid. Now what happens? This ammonia can accept one hydrogen atom and become ammonium. So you see here also, when a hydrogen atom is produced, hydrogen ion is produced by this reaction. This will be by the pump, proton pump. It will be eliminated here. It will be secreted here. Now it will combine with this deaminated ammonia to form NH4, and this way it will be excreted. So isn't it? It is very easy. Very simple. So it is not so tough. So that's in that way the kidney, by this three system, can control acid-base balance. Okay. Now what is acidosis? What is alkalosis? we'll find acidosis are of two types. One is respiratory, means this acidity is because of respiration. How it can produce acidosis? By suppressing breathing. Accumulation of carbon dioxide means more production of hydrogen. Okay, and suppression of respiration can happen in various conditions. Okay, so this is respiratory acidosis. And there is also metabolic acidosis. Okay. Alkalosis means opposite. pH will be what? Up. pH will be high. Okay. So in this condition, when you are going beyond the range, 7.45, beyond this, then the condition is alkalosis. Okay. Normally PCO2 in the blood, partial pressure of carbon dioxide. What is partial pressure? What is the atmospheric pressure? How partial pressure? What do you mean by partial pressure? So here is what is the atmospheric pressure, barometric pressure? Say 760 millimeter of mercury. This concept you must have, otherwise what is this partial pressure, partial pressure? (760) minus water vapor. Because this pressure is 760 millimeter mercury pressure. Who are exerting together? There is contribution of carbon dioxide, there is contribution of oxygen, contribution of water vapor. Okay, if I subtract these, suppose it is 60, so remaining 700 millimeter of Hg. Who is creating this pressure? Carbon dioxide and oxygen. So here how much will be the partial pressure of oxygen? How much will be the partial pressure of carbon dioxide? It depends on what is the concentration of carbon dioxide here. What is the carbon dioxide concentration of oxygen here? If you take the 100 ml of air from here, how much oxygen you will take? 21%. So this 21% of oxygen is exerting some pressure to create 760. What is its role? 20. What is the concentration? 21% or 20%. That means 20% of this, 20% of this is contributed by oxygen. Similarly, this is the partial pressure of oxygen. Similarly, partial pressure of carbon dioxide. How much it is? What is the percentage here? 0.03. So, multiply 700 by 0.0, you will get the partial pressure of carbon. So, this is what we mean by partial pressure. You understood this? Right? Okay. So, this is simple. I am having already time is over. So, very simple. What is acidosis? You just go through, you will find it. And you may sometimes it may be asked, what is the comparison? This respiratory acidosis, what is the difference? So you try to learn it, short question you can expect from here. Alkalosis, acidosis and their types, and what are the symptoms. So you have to learn the symptoms, you have to learn the two types. In case of metabolic acidosis, what are the symptoms? In case of respiratory acidosis, what are the symptoms? Similarly, for alkalosis, you have to run what are the causes, what are the symptoms, how it can be managed. Okay, management always, if possible, physiologically, not by any drug. You don't know about drug now. So physiologically, it can, whether it can be managed or not, that you can right there. And another important question. This is also comes every 2-3 years. What is this? This one. Go through it. Try to understand. Read it. Very simple. Don't write your answers from Guyton, particularly for Anion Gap. Some other books. In all books it is not there. So go through Guyton. I should not to go through Shambulingam. Not to go through Shambulingam. Guyton is okay. Okay, or Devashish Chakravarti is also okay. Devashish, what is his name? Anyway, so how Anion Gap is measured? Summation of this, you got the report of electrolytes. Okay, so you got sodium concentration, you got potassium concentration. Sum it. You also got bicarbonate concentration and chloride concentration. Sum these two cationic, anionic concentration. Sum of these two and sum of these two. This will be high relative to this. So subtract this value from this and you will get a value which will be between 3 millimole per liter to 11 millimole per liter. This is liter. Millimole per liter. Okay, now what does this, when it decreases, when it increases, what does it mean? You have to explain. Okay, so this is about Anion Gap. Again, you got the idea about all this? So prepare it today. Okay, so lot of questions you got, important questions you got. Now another important will be definitely, I think in the next class, it is mixturation. And that is also another important question. The reflexes, the mechanism of mixturation, that is also important. And remaining part, different these clearance values. So these are our renal function test and kidney transplantation. So these are some these 3 marks questions.